Chapter 21, Viruses. This video lecture was recorded on January 13, 2021, in the midst of an ongoing COVID-19 pandemic. I will make several references to this virus since it is so relevant to our lives during this time. The information that I use in this chapter regarding this virus may change as our understanding of it changes. Regardless, keep wearing masks and get your vaccine when you are able. The diversity of life begins with viruses. Although it's a paradox to even call them alive because they are non-cellular parasites that infect living cells. This is the coronavirus. However, this name represents a class of viruses with crown-like spikes that include many cold viruses, the SARS virus that ravaged Southeast Asia in the early 2000s, and the MERS pandemic in the early 2010s. Its real name is the Sudden Acute Respiratory Virus 2, or SARS-CoV-2, and it was the bane of our existence in 2020 and 2021. Viruses are perhaps the oddest things on the planet. While they possess genetic material, except prions, which I will discuss later, they replicate. They are acellular and have no growth or metabolism. While they reproduce, they can't do so without taking up residence inside of a living cell. Therefore, viruses are obligate parasites. This means they can only replicate inside a host cell. The first virus was discovered by Adolf Meyer in 1886, and that was the tobacco mosaic virus, which, as the name suggests, infects tobacco plants and their relatives. Although the scientists couldn't see the mosaic virus at the time, they were able to detect it using the filtration processes used to extract it. Viruses are so small that most can't be viewed without the aid of an electron microscope. Take a look at the right picture. This contains human epithelial cells with smaller E. coli cells shown as black rods. If you zoom into the surface of an E. coli cell, you can see the virus on its surface depicted in the left picture. That's how small they are. Think of small viral particles as virions. These are usually not inside of a host cell and therefore are not harmful yet. While we understand how modern viruses mutate and adapt, which is why you need an annual flu shot, by the way, we know very little about the origins of viruses. Virologists, which are virus scientists, agree that viruses do not share a common ancestor like living things do, so there's three hypotheses proposed. First is the regressive hypothesis, which states that viruses evolved from free-living cells or intracellular parasites of prokaryotes, almost like they de-evolved. Then there's the progressive hypothesis, which says that viruses originated from RNA and DNA molecules that escaped from a host cell. And finally, the third hypothesis is that self-replicating hypothesis, in which viruses originated from self-replicating entities similar to transposons, or jumping genes. Regardless, understanding viral evolution will make it easier to develop treatments against the diseases they cause in humans, wildlife, and agriculture. As mentioned previously, viruses are acellular, meaning there's no membranes or ribosomes or organelles. This is why there are so few antibiotics or antiviral drugs against viruses. They lack the typical targets, and second, viruses live inside your own host cell. So if the antibiotic were to kill the virus, it would ultimately kill the host cells. The basic anatomy of a virion is a nucleic acid core of DNA or RNA. There's never both inside at the same time, and it is surrounded by a protein capsid. Sometimes there is an outer envelope made up of proteins and phospholipids that derive from host cells. Those with enveloped are called enveloped viruses, and these are pretty common in animals, while those without an envelope are called naked viruses and are common in plants and bacteria. Bacterial virions are called bacteriophages, and they are perhaps the most bizarre looking things. Viruses evolved alongside their host since they rely on their host for replication. Viruses are quite diverse in shape and size, but can mainly be classified into one of four shapes. Helical viruses, such as the tobacco mosaic virus, are long and cylindrical and mainly infect plants. Icosahedral viruses have a spherical-like shape and are found in polioviruses and herpes viruses. Enveloped viruses have membranes and derive from the host cell's phospholipids and or proteins. HIV and SARS-CoV-2 are examples of this. Most enveloped viruses infect animal cells. And another common one are head and tail viruses, which infect bacteria and have a head similar to an icosahedral virus and a tail shaped like a helical virus. Scarlet fever is caused by a normal Streptococcus bacterium found in your throat being infected by a bacteriophage of this shape, and that causes the bacterium to start producing toxins that can infect us. Viruses make use of viral receptors on host cell surfaces so they can dock and insert their DNA into the host cell. While these viral receptors actually perform other functions for the host cell, viruses have produced attachment structures like spike proteins or tails that can fit into the same receptor sites. Understanding how the virus docks and injects its genetic material is a big part of learning how to fight and treat infection. The SARS-CoV-2 virus, also known as the coronavirus, is an enveloped virus that has spike proteins that attach to the angiotensin-converting enzyme receptors found in our lungs, arteries, heart, and kidneys. Now, the purpose of these angiotensin-converting enzyme receptors is to help with our blood pressure in our body. Coronavirus has similar spikes that can be used to dock and enter our own cells, especially within our lungs. Viruses have either DNA or RNA, never both, and this genome contains information for building proteins that the virus can make and not get from its host. The DNA or RNA could be double or single-stranded, circular DNA or linear. Viruses could also have a single nucleic acid or several segmented ones. In fact, influenza viruses have segmented nucleic acids, making it very difficult to vaccinate against. The best we can do is predict which strains of seasonal flu will be the deadliest and develop vaccines against those. DNA viruses, as the name suggests, contains DNA, and the viruses utilize the host cell's machinery to replicate, transcribe, and translate viral genome and viral proteins. Examples of DNA viruses are chickenpox, hepatitis, and human papillomavirus, also known as HPV. RNA viruses contain only RNA as their genetic material. These viruses must replicate their own genomes using their own RNA polymerase enzymes. Some RNA viruses are termed retroviruses, as they can perform reverse transcription, in which their RNA can be used to make DNA. Examples of RNA viruses include the coronavirus, also known as SARS-CoV-2, influenza, and HIV. HIV is known as a retrovirus. Point of note, retroviral infections are easiest to treat since humans can't perform reverse transcription. Many of these antivirals target this process. As you can guess, classifying viruses is really difficult, since there's no fossil record, nor there's even an agreed-upon hypothesis as to where these guys originate from. Systematics utilizes evolutionary relationships to form groups in prokaryotic and eukaryotic life forms, but we can't really use those same tactics with viruses. Therefore, scientists have devised different classification schemes over the years. Historical classification methods mostly focused on morphological structures such as RNA or DNA, single or double-stranded, linear or circular, and segmented and non-segmented DNA. Some virologists have also placed them into groups by capsid types such as naked, icosahedral, enveloped icosahedral, enveloped helical, naked helical, and complex. Well, since the 1970s, virologists have used David Baltimore's system of classification, which places viruses into 7 groups based on how the mRNA is produced during the replicative cycle of the virus. They are Group 1, which have double-stranded DNA with mRNA produced by transcription-like cellular DNA using the host cell's enzymes. So, in other words, they replicate a lot like eukaryotic and prokaryotic cells do. In Group 2, they just have single-stranded DNA with messenger RNA made by converting the single-stranded DNA into double-stranded DNA before using the host cell's enzymes for transcription. And the plus and the minus refer to the complement nucleic acid strands. So if you have a plus strand, the opposite strand is the negative strand. In group 3, they have double-stranded DNA, but the strands separate, with one of the strands used as a template to make messenger RNA. In group 4, they have single-stranded RNA with the positive polarity, meaning that the RNA can actually serve directly as mRNA. So this group has a positive single-stranded RNA, and this can be used to make the opposite strand negative single-stranded RNA, or they could just immediately make amino acids as needed. So anytime you see positive single-stranded RNA, it can immediately be used as mRNA, and it will serve as that purpose. In group 5, they have single-stranded RNA with negative polarity, meaning that it needs to build the complement positive single-stranded RNA before it can act as mRNA and make amino acids. In group 6, they have 2 copies of single-stranded RNA genomes that must be converted using the enzyme reverse transcriptase. This takes the single-stranded RNA and uses it to build the complement single-stranded DNA. Afterwards, this single-stranded DNA is then inserted into the host genome where transcription of mRNA happens. And finally, in group 7, they have partial double-stranded DNA genomes and make single-stranded intermediates that act as messenger RNA. And they can also use the mRNA to undergo reverse transcription, inserting that DNA into the host cell's DNA. Up next, we're going to look at virus infections and host treatments of viral infections, and in viroids and prions.